Good morning, everybody, and welcome. I'm the daughter of earth and water and the nursling of the sky. I change, but I cannot die. I'm really confused whether this is a version of a cloud or COVID-19. At present, we are really under stress condition, but of course we are trying to overcome it. I think this webinar platform or online program is one such way to overcome the situation. But plants have some different mechanism to overcome their threats. That is why today we are here with our webinar topic, microRNA key players of plant responses to abiotic threats. I must mention that this webinar program is jointly organized by the Department of Botany and IQAC of Navadip Vidyasagar College, and Dr. Satidas Shur and Dr. Shubhadeep Chakraborty will chair the session. Today we have our respected principal sir, Dr. Shapon Kumar Rai, in this session. We never forget his contribution, his encouragement and continuous support during the entire tenure of our webinar program. So thank you, sir, for being with us. Really, today we can't be here without you. At the same time, I must mention the names of some of my esteemed colleagues, Professor Dipanjan Ghosh, Professor Rajkumar Mondol, and Syamalendu Dash for their continuous effort and support. Thank you all. I must place my thanks to our honorable speaker, Professor Jayed Hossain of Kalyani University. Thank you, Professor Hossain, for sharing time with us. I know you are a very busy person, but somehow you can manage the sheet for us. So thanks once again.
Thank you.
So we are late by 1 minute, so I don't want to waste my time. Now I am requesting our respected principal sir, Dr. Shapon Kumar Rai, to deliver his welcome message on behalf of Navadip Vidyasagar College. Sir, please.
Thank you, Dr. Sengupta. Can you hear me?
Yes, sir. You are clearly audible.
At the inception, I actually express my thanks and gratitude to the Department of Zoology, Botany, who and in collaboration with the IQAC, they are organizing this web seminar on microRNAs, key players of plant responses to biotic stress. We have with us our prominent, eminent, distinguished speaker, Professor Dr. Jayed Hossain, who is actually the head of the department, Department of Botany, University of Kalyani. We are grateful to him because he has accepted our invitation, and he is also happy to be with us and to deliver his speech on the same topic. I think his scintillating lecture speech, his speech on the topic will enrich the participants actually. If the participants be happy, if the participants be contented, then I will think that the web seminar will be a successful one. So thank you again on behalf of Navadip Vidyasagar College, from the Department of Botany and from the all departments of Navadip Vidyasagar College. Thank you again for accepting our invitation and for giving his consent to be present with us, for being with us. Thank you again, Dr. Jayed Hossain.
Thank you.
Secondly, I express my thanks and gratitude to the participants who have joined this webinar. We know the participants are the source, our strength. Our strength, if the participants are less, then for which we are going to organize such seminar, it will not be a successful one. So the participants be happy, participants be contented by the valuable speech, by the scintillating speech to be delivered by our Professor Dr. Jayed Hossain, who is associated with the University of Kalyani and the head of the department. I think he would be person who will give his very valuable speech, which will make the participants satisfied. So all the participants on behalf of Navadip Vidyasagar College from the Department of Botany, I express our thanks, gratitude to all participants who have joined this program, web seminar. Thirdly, sometimes I forget to mention the name, site name, because a number of teachers are associated with this web seminar. Last day we have missed to mention one name, who is a spirited teacher and a devoted teacher. I could not mention her name. Dr. Sati Dasur, head of the Department of Botany. Perhaps she'll be satisfied today, happy today, because at the very beginning I have mentioned his name. Dr. Sati Dasur, who is a convener of this webinar, and also the IQAC coordinator, Dr. Bhaskar Chatterjee is always with us. Dr. Bhaskar Chatterjee, Internal Quality Assurance Cell coordinator, he is always with us, and we thank him for extending his cooperation in organizing this such type of seminar. Thank you, Dr. Bhaskar Chatterjee, who is the Department of Chemistry, Navadip Vidyasagar College. Thirdly, we actually congratulate the members, all the members of the organizing committee. Uh, we know that the members, perhaps most of the participants who have joined from outside uh, Navadip areas. Hmm. We know, actually we are very much grateful, but because they are the actually gems of Department of Botany. Dr. Subhadip Chakraborty, Dr. Kaushik Sengupta, he is always with us.
Thank you,
Mm,
sir.
Dr. Damayanti Bhattacharya. So, and Dr. Sati Dasu, they are actually the wealth of or treasure of our college. Thank you again, the members of the organizing committee and the faculty of the Department of Botany. Thirdly, who actually has been playing a very important role, pivotal role from behind the screen uh, in order to make this webinar successful. Uh, Dr. Professor Dipanjan Ghosh, who is from the Department of English. Professor Rajkumar Mundul, who is from the Department of Physics. And our beloved librarian, who is actually dear, uh, Mr. Amanendu Das. These three are always with us, who are playing a very important role from behind the screen. So they are the members of the technical support. On my behalf, I my special thanks uh, to these three teachers, Professor Dipanjan Ghosh, Sri Amolindu Das, and Professor Rajkumar Mundul. At last, I express my thanks and gratitude to our honorable president, governing body, Sri Biman Krishna Saha, who is our source of inspiration. He is always with us. Whenever I call him, he actually says me that to uh, to arrange such type of seminars. So thank you, sir. In spite of his absence, but he has given me a liberty, a tons of liberty, so that we can continue our webinar. Thank you all, especially Dr. Zahid Hussain, who has given his consent to be with us as a speaker and deliver his speech as a speaker. Thank you again, Dr. Zahid Hussain, Professor, Department of Botany. Thank
Thank
you
you.
from the core of our heart from Navadip Vidyasagar College, all teachers, all non-teaching staff, all students, and the participants. We thank you again, sir. Over to Dr. Kaushik Sengupta.
Thank you, sir, for nice delivery. and you, in brief, covers all the arena required today. Now I am requesting Professor Dr. Shati Dashu to introduce our honorable speaker, Professor Hoshen, and address the participants in brief. Dr. Shu, please. Can you hear
Okay.
me?
Yes. Am I audible?
Ah, you can continue.
Okay. Okay. Thank you, Dr. Sengupta. Very good morning. On behalf of the organizing committee of the webinar, I welcome all the delegates, speakers, and participants. We're excited for organizing such an academic discussion through online platform for the first time. We are thankful to our Honorable GB President, Sri Biman Krishna Saha, for his continuous support. And our special thanks to our Principal Sir, Dr. Shapon Kumar Roy. We thank him for his continuous encouragement, and to be honest, this webinar is his brainchild. My special thanks to my colleagues, Dr. Kaushik Sengupta, Dr. Damayanti Bhattacharya, and Dr. Shubhodip Chakraborty. Their constant support and knowledge enable me to be here. And also thanks to Suraj Bani, our departmental non-teaching staff. Our IQAC coordinator, Dr. Bhaskar Chatterjee, also collaborated with us. We are thankful to our academic convener, Sri Dipanjan Ghosh, for his continuous support, convener of Technology Upgradation Subcommittee, Sri Rajkumar Mondal, and Amolendu Das, our beloved librarian. And last but not the least, we are thankful to our technical supporting team for their great effort. Today, we have with us Professor Dr. Jahid Hussain, our honorable speaker. Let me introduce him. Dr. Hussain currently is the head of the Department of Botany, University of Kalyani, and coordinator, Genome Science, School of Interdisciplinary Studies, University of Kalyani. He is recipient of several prestigious national and international awards and fellowships. He was a visiting researcher of Ecophysiologia, Biotechnologia, Universitat Jammu, Spain. He was an invited fellow of Better Opportunities for Young Scientists in Chosen Areas of Science and Technology for the Department of Science and Technology. He was also an invited fellow of Japan Society for the Promotion of Science for the Institute of Crop Science, Tsukuba, Japan. Dr. Hossain's research interests include small RNAs, transcriptomics, and proteomics. His group investigates plant responses towards different environmental challenges at physiological, biochemical, and transcript and proteomic levels. Currently, his team is engaged in exploring the role of miRNA in abiotic responses. 3 decades ago, small RNAs of less than 30 nucleotides were not considered to have any biological role. Then a few discoveries came out and changed the game totally. Icing on the cake happened when in the year 2006, Fire and Mello were awarded Nobel Prize for their discovery of RNA interference gene silencing by double-stranded RNA. In today's world, RNAi is one of the most active fields of biological research. In RNAi, 21 to 24 nucleotide long RNA along with associated protein downregulates gene either by mRNA degradation or by translational inhibition. The biological role of such RNAi is host defense and regulation of gene expression. Scientists have applied RNAi in different fields like treatment of cancer to production of genetically engineered Arctic apple. miRNA is one of such small RNA which play critical roles during plant development and in response to abiotic stress like drought, salinity, oxidative stress, UV radiation, hypoxia, aluminum toxicity, etc., and nutrient homeostasis. Our honorable speaker, Professor Dr. Jahid Hussain, will deliver his lecture on microRNAs, key players of plant responses to abiotic stress. Thank you, Dr. Hussain, for being with us. And this knowledge will help us to come up with crops adapted to post-climatic change era. Thank you.
Thank
Dr.
you.
Sengupta, please switch over the microphone to our honorable speaker.
Thank you, Dr. Shur. You have sorted out all the importance of miRNA. So far, my knowledge goes, in plant states, tolerance is genetically controlled, and MIR398 plays a significant role. Dr. Hussain will say, I think. And I am actually, Dr. Hussain is known from his since student life to me, and today he will get good enough from his end. I hope so. So I don't want to waste any more time. I'm requesting Dr. Hussain to start his presentation. Dr. Hussain, please.
Yeah. My voice is audible?
Yes, you are
Hello?
clearly audible.
Okay. So very good morning to all of you. It's been a pleasure for me to deliver a lecture in this webinar. And I'm thankful to Dr. Shathi and Dr. Kaushik Sengupta for nicely introducing myself. And first of all, I would like to thank Dr. Shapan Kumar Roy, Principal, Navadip Vidyasagar College as Chairperson, and Dr. Shathi Das Shur, Head Department of Botany as Convener, Dr. Bhaskar Chatterjee as IQAC Coordinator, and Dr. Shubhadeep Chakraborty, Dr. Kaushik Sengupta, and Dr. Damayanti Bhattacharya as the members of the organizing committee for inviting me. So without wasting time, I would like to start my lecture. Shall I? Okay. Just a minute. Okay.
Okay, we are waiting. No problem.
Okay. So now I have to share. Okay.
Share interesting.
Present. Okay, just a minute.
Present
Enter
now,
screen.
share interesting. Select
Yeah.
and share.
Yeah.
Yes, it's coming.
Yeah.
Manjur Alam, participant Manjur Alam,
Yeah.
please stop presenting. Otherwise, we have to remove you from the seminar. Manjur Alam, Steve,
So now-
please stop presenting.
So
Professor
now
Hussain, please
the
wait.
slide.
Please
Yeah.
wait for
Okay.
a short while.
Okay.
Someone is presenting. Manjur Alam. Okay. Professor Hussain, now you can proceed.
Okay. You can see the slide. Yeah.
Slide. Okay. It's okay.
Okay. It's okay. Okay. So myself, Dr. Zahid Hussain.
Presentation is muted. Presentation is muted. Please open the slide in same window.
You cannot see my slides.
It is at the corner. So please pin it in the center.
Is okay.
Still running. I think Dr. Hussain, it will be better if you go back and start representation. Okay.
Okay.
Present now. And all participants, please be with us following all instruction given by the Dr. Shubhadeep Chakraborty. Don't try to present now. Professor Hussain is our honorable speaker. We will present soon.
Now you can see.
It is coming. Yes, it's now okay.
Is okay. Okay.
Perfect. Perfectly.
It's perfect. Okay. Okay. Shall I
And
start?
okay. And
Okay.
you can start.
Okay. Myself, Dr. Zahid Hussain. Currently, I am associated with the Department of Botany, University of Kadani, and I have been working in the field of plant abiotic stress. You are already aware the today's topic of my lecture is microRNAs, the key players of plant responses to abiotic stress. So before going into details, we should know what is called stress. So any unfavorable condition that basically limits the crop productivity or destroy the biomass is referred to as a stress. So plant stress, we can classify plant stress into two categories. One is a abiotic stress, another one is a biotic stress. So abiotic stress, it includes salinity, water stress. The water stress, it may be an excess of water, that is called the flooding or the submergence, or it may be a scarcity of water, that is a drought, or it may be a temperature stress. It may be a very low temperature, it may be a heat, or it may be a frost, or sometime nutrient imbalance. Sometimes it creates a stress. Hello, hello.
Dr. Hussain, your slides are not visible. So go
Why?
to present now mode again. And then share your screen.
Is it okay?
Yes, yes, visible.
Visible?
Yes.
Is visible? Hello.
Yes, Dr.
Okay,
Hussain.
okay,
Yes.
okay. I will
Yes,
continue.
yes.
Okay.
It's visible.
So again, some nutrient imbalance, it sometimes creates stress for the plant, and you can say the light, different kinds of light. It may be a highlight, it may be a UV light, or it may be even a darkness. Basically a stress for the plant. And apart from that, there is a very important group, that is the heavy metal stress, which includes the application of, or you can say the stress effect due to the presence of high amount of arsenic, cadmium, or aluminium or copper. And apart from this, there is another group of stress that is called the nanoparticle stress, which is basically a new class of pollutant. And the another category, that is the biotic stress, it basically includes the infection caused by the bacteria, fungi, or virus, or even insect, nematodes, or any other pathogens. So being sessile, plants are constantly exposed to different types of stress. It may be heat stress, it may be cold or flooding or drought or salinity. So plants, like other aerobic organisms, they require oxygen for the efficient production of the energy. So during the reduction of oxygen to the molecule to the water, a series of reactive oxygen species are formed, namely superoxide radical, hydrogen peroxide, hydroxyl radicals, and singlet oxygen. So when a plant experiences any kind of stress, the delicate balance between the formation of the reactive oxygen species and the quenching activity of the antioxidant get disturbed. And as a result, the excess ROS generated under the salt stress, it basically unbalances the cellular redox system in favor of oxidized damage or oxidized form, resulting oxidative damage to lipids, proteins, and nucleic acid. So during the course of evolution, plants have evolved a very robust antioxidant defense system, which basically allows scavenging of ROS and protection of plant cell from the oxidative damages. And these antioxidant defense system, it basically comprises of enzymatic system and the non-enzymatic system. So enzymatic system basically includes some enzymes of ascorbate glutathione cycle, namely superoxide dismutase, that is called the SOD, the ascorbate peroxidase, the monodehydroascorbate reductase, dehydroascorbate reductase, or glutathione reductase. And the non-enzymatic system, it basically comprises of different antioxidant molecules, for example, glutathione, then ascorbate, then carotenoids, vitamins, and the flavonoids. So all these are the good quenchers of the reactive oxygen species. So this is basically, you can see that most of the cellular compartments, they have the potential to become a source of or ROS. So the slide showing the interconversion of the reactive oxygen species starting from the molecular oxygen to the water, and each stage there is a 1 electron reduction. So you can see that the molecular oxygen is basically converted to a superoxide radical, and then further it is converted into hydrogen peroxide and hydroxyl radical, and like this. So this is basically, you see, this is the ascorbate glutathione cycle that is being operated in most of our plant cell organelles, and it comprises of 5 antioxidant enzymes. As I mentioned, this is the superoxide dismutase SOD, and the ascorbate peroxidase APX, then MDR monodehydroascorbate reductase, then dehydroascorbate reductase, and glutathione reductase. And apart from these 5 antioxidant enzymes, there are 2 antioxidant molecules are there, both that is glutathione and ascorbate, and these two are present in 2 forms. One is a reduced form, another one is oxidized form. So this ascorbate glutathione cycle, they basically play a very vital role in maintaining ROS homeostasis within the plant cell. So you know, these are the different high throughput omics techniques which are being currently used to decipher the underlying molecular mechanism of plants' responses to various stress. It may be a genomics, it may be transcriptomics, it may be proteomics, metabolomics. So today we will focus a specific omics technique that is called microRNA. So before starting, how you know that a plant is experiencing a stress condition? This is very much important question. So you see this this picture showing some symptoms of here it is heavy metal stress. So symptoms of cadmium toxicity. In the left panel, this one is basically a healthy plant. It's basically a maize seedling. It's a 7-day-old maize seedling. And this plant, this one, the T1, is basically a cadmium stress maize seedlings. What we did, we basically imposed the cadmium stress to the 7-day-old maize seedling. We imposed 100 micromolar cadmium, and after 4 days of treatment, this particular photograph was taken, and you see the plant is showing the symptoms of clear symptoms of cadmium toxicity. And you see these symptoms basically includes the leaf tip yellowing followed by the drying, and the symptoms first appear in the first leaf, then in the second leaf, and then in the third leaf respectively. So here you can see this is a comparative photograph of the first leaf. So this is the control leaf. This is basically a very green healthy leaf, whereas in case of the cadmium stress leaf, this particular first leaf is you can see this is completely dried up due to the cadmium treatment. And again, the second leaf, this one is a healthy second leaf for the control plant, whereas this particular second leaf, it has already started the symptom of cadmium toxicity. You can see the leaf tip yellowing has started, and the leaf has already started the drying symptom. So these are some of the common symptoms of heavy metal stress. So as I mentioned that whenever a plant is experiencing any kind of stress, the delicate balance between the formation of the reactive oxygen species and the quenching activity of the antioxidant get disturbed. So there is an excess formation of the ROS. So how you can detect the ROS? So there are 2 common histochemical staining method. One is called the nitro-blue tetrazolium, which is very specific to the superoxide radical detection. So what we did, this is basically a control leaf, and these are the cadmium stress leaf. So after doing the NBT staining, chlorophylls were removed, and then you got to know that you can find these blue color stripes are here, and this is the enlarged microscopic view. So what happened, the NBT basically reacts with the superoxide radical, and it forms an insoluble formazan. And that insoluble formazan basically get trapped within the cell, and you can find this blue color deposition. So these deep blue color spots basically are indicative of the superoxide deposition. And similarly, there is another stain that is called the Dept, and this Dept is basically a specific stain for the hydrogen peroxide. So again, after the treatment with Dept and after the removal of chlorophyll, you can find this brown spot for these cadmium stress leaves, and this is the enlarged microscopic view. So presence of these brown spots basically are indicative of the hydrogen peroxide deposit. So using these common histochemical staining, one can understand the whether there is an enhanced ROS generation under the stress or not. So usually when plant is experiencing a stress, it is obvious the rate of the production of this ROS is basically much higher as compared to the scavenging capacity of that plant. So this is another there are some fluorescent dyes are there. One can use those dyes. A very common one is DCFDA, another one is a DHE. These DCFDA is basically a hydrogen peroxide specific stain, the fluorescent stain. So you see this is this one T0 is basically a control maize root tip, whereas this T1, this is basically a cadmium stress root tip stained with the DCFDA. And under the fluorescent microscope, you can see that the bright green colors are there, and the intensity of the green color here is much higher as compared to the control. That means, which indicates a higher production of the H2O2 under the stress condition. Similarly, you can use another stain that is called the DHE, this dihydroethedium, that is basically a specific to the hydroxyl radical specific stain. So here you can see, as compared to the control, the intensity of the red color is much higher, which again indicates the presence of high amount of, or formation of high amount of superoxide under the stress condition. So all these whatever the mode, whether it is a histochemical staining or stained with this fluorescent dye, you can see that there is an excess generation or loss, or you can see there is an oxidative burst under the stress condition. Even one can measure tissue hydrogen peroxide level, and by a normal spectrophotometric method, here you can see that as compared to the control, there is a sharp increase in the H2O2, tissue H2O2 level within the stress leaves. So one can even identify histochemically all with a spectrophotometric method the amount of ROS that is generated within the tissue under the stress condition. And another very important parameter is, this is called malondialdehyde. This is basically an indicator of the membrane damage, or you can say the lipid peroxidation. So what happened that here you can see that as compared to the control, when we measure the MDA level, it's sharply high. So with the increase in the stress level, there is an increase in the MDA concentration, tissue MDA concentration, which basically indicates the stress effect on the membrane, that is the, you can say, the higher lipid peroxidation. Or one can study the expression of antioxidant genes or the defense-related genes using either a QRT PCR. Here, this panel is for the QRT PCR, or one can do a semi-quantitative semi-quantitative RT-PCR. And here you can see that we studied the 5 antioxidant enzymes of the ascorbate glutathione cycle, and there is a differential modulation of these antioxidant gene transcript. So which basically indicates that higher level of ROS is formed, and plant is trying to mitigate those ROS level so that the plant can survive the stress. Even we used to validate these transcriptome data with the protein expression data. Here you can see, last slide we have shown the the transcript level of GR. Here you can see this is the protein expression level of these antioxidant protein. For example, this is a GR protein expression. You can see is 54 kilodalton, like that. It's CAPX protein expression, copper zinc SOD expression. So all these are the validation of your transcript data. So far what we have understood is what is called a stress, what are the different forms of stress, and what are the symptoms of stress, and what are the different parameters one should study to evaluate the plant response under the stress condition. So the big question is, who are the key players that basically regulate the gene expression at a post-transcriptional level? So broadly, we can classify the RNA transcript into 2 broad groups. One is a protein coding mRNA, another one is a non-protein coding RNA transcript. So the protein coding mRNA, we know these specify the polypeptide that encodes the protein. Whereas there is another group that is called the non-protein non-protein coding RNA, which do not specify any polypeptide, and they remain untranslated. So these non-coding RNA transcripts, basically you can classify into 2 categories. One is one is called regulatory RNAs, and another one is a housekeeping RNAs. So housekeeping RNAs, our examples are, we know these are the tRNA, rRNA, telomerase RNA, which are involved in the housekeeping function. So you know tRNA, rRNA, those are always involved for the protein synthesis mechanism. Whereas the regulatory RNA, it includes the miRNA, that is called the microRNA. Another one is a short interfering RNA, that is the siRNA, and another one is a PV interacting RNA, that is called piRNA. So these non-coding regulatory RNAs, these group, These are basically involved in the post-transcriptional gene silencing. So what are microRNAs? So from the name, it is very much clear to you that micro means is very small in size. You know, in your mobile, there is a micro SD card. That means the card size is much smaller than the normal conventional card size. So similarly here, the micro means it's a very small size. It is usually 19 to 24 nucleotide in length. These are highly conserved and these are non-coding. As I already mentioned, that they do not code for any polypeptide, but they regulate gene expression at a post-transcriptional level, either by, there are two mechanisms, either by the mRNA degradation, that is called the cleavage, or the translation repression of their target, that is basically the inhibition of the protein synthesis. So these microRNAs are usually associated with the diverse biological processes that Shati has already told, that they are involved in organ development, organ maturation, cell proliferation, and flowering time. However, there are so many papers coming out in recent times that microRNAs are basically play potential role or pivotal role in plant responses to various abiotic stresses. So this is the first described microRNA, that is called the Lean4, and Rosalind Lee in 1993, she first discovered this microRNA, and it was discovered in the Rhabditis elegans, is basically a soil nematode, and Lean4 was found to be silence another gene, that is the Lean14. So you should remember the first described microRNA, it was in 1993, and it was the name was Lean4. And Rosalind Lee, she first discovered this Lean4 microRNA. So what are non-coding? NC means non-coding. So what are non-coding microRNAs? So there are two groups, as I mentioned, apart from the PV, that is the miRNA. Another one is a short interfering RNA, that is the siRNA. And both are non-coding. That means they do not code for any polypeptide, but they share some common homology first. They both are involved in gene silencing, whatever the mode, I will tell you later. And the second one, both are small in size. MicroRNA is around 19 to 24 nucleotide, whereas the siRNA is around 22 nucleotide long. And the microRNA, usually they are encoded by the nuclear genes, whereas the short siRNA, they are basically derived from very long double-stranded RNA molecules. So first we should know the biogenesis, the target, and the mode of action of the microRNAs. So whatever I'm going to show is all about the animal microRNA biogenesis system. Later I will tell you the plant system mechanism. So the microRNAs, these are encoded by nuclear gene. That means the genes are present within the nucleus. So the microRNA genes are present in almost all multicellular eukaryotes. Even some unicellular eukaryotes, they have these microRNA genes, for example, Saccharomyces cerevisiae, and even some transposable element, they can carry the microRNA genes. And you know, in the case of human, these microRNA genes are dispersed throughout the all human chromosome except for the Y chromosome. And these microRNA genes, as I mentioned, they are present within the nucleus. So they may be present alone. That means they may be present in between the two protein coding genes, or they may be present within a protein coding gene. If it is present within the protein coding gene, it should be present in the intron region of a protein coding gene, or sometimes it has been also found that they are present within the exon region of another non-protein coding gene. So whatever their status or the location, the microRNA genes is first transcribed by the RNA polymerase II, you can see, and it gives rise to a very long primary microRNA. And it has a 5' cap and a 3' poly A tail. You know, these are the features of eukaryotic mRNA. And so these primary microRNA, it has some sequence homology. So it basically pairs and forms a secondary structure that is called the stem loop structure. So this primary microRNA is formed within the nucleus, and then it is processed by a protein called DROSA, and there are certain accessory proteins are there, which these DROSA basically, it's basically a double-stranded RNA-specific endonuclease. So it basically creates staggered cut at the base of the stem, and it results in the 2-nucleotide overhang at the 3' region. So this shorter microRNA is known as precursor microRNA. Basically, the primary microRNA is around 70 nucleotide long. Then it is further processed by DROSA, and it forms a precursor microRNA. So once precursor microRNA is formed, it is readily transported from nucleus to cytoplasm through this nuclear pore, and there is one protein called exportin 5 that basically helps the transport of these precursor microRNA from nucleus to the cytoplasm. So this is basically the processing of the primary microRNA to the pre-microRNA. So here is, as I mentioned, DROSA is basically a double-strand RNA-specific endonuclease, and it causes a staggered cut. Here is one cut, here is another cut. So it results in this 2 nucleotide overhang at the 3' end. And very interestingly, you can see within the stem structure, there is a bulge. That means for these two nucleotides, there is no complementary sequence in the other strand. So this is very unique of this microRNA strand. So once the precursor microRNA is within the cytoplasm, it is further processed by another protein that is called the dicer, and here it is present in the yellow color. This dicer along with the accessory protein that basically binds at the base of this loop, and it creates another staggered cut, so that now a microRNA duplex is formed. So this duplex is consists of one microRNA strand, which is known as guide strand, or why it is guide? Because it basically directs the RNA silencing. And the another strand with a bulge is basically known as the passenger strand, which is eventually will get dissolved. So now there is another protein that is called the AGO1, which is also a slicer protein, which along with the accessory protein will go and bind to this microRNA duplex, and it forms a pre-microRNA induced silencing complex, I think pre-MI risk. Now the AGO1 is basically an endonuclease that cleaves this passenger strand, this strand, and the helicase, which is associated with the accessory protein, that basically unwinds the two pieces and they dissociate from the complex. So now this is called the mature MI risk, and this MI risk is consist of a mature microRNA, this AGO1 protein, this dicer, and all the accessory proteins. So now very interestingly, this mature MI risk will go and bind to their target mRNA or the target mRNA transcript, and they usually bind at the 3' UTR region of the target mRNA, and here you can see the pairing is imperfect. Here you can see a bulge, that is the mature microRNA, the green one, and this is the target mRNA. So since the pairing is imperfect, it basically represses the translation of these target mRNA. So in this way, it basically silence the gene expression, because there would be no translation, there would be no protein product. So since this repression occurs after the transcription, after the formation of the transcript, it is known as post-transcriptional gene silencing. So this is the processing of precursor microRNA to the RNA duplex with the help of the dicer. It basically cut this loop part. So precursor is converted to the RNA duplex, as I mentioned. So this is very important slide. You can see, as I mentioned, the mature MI risk will go and bind to the 3' UTR region of the target mRNA. Why it binds to that? Because they have the sequence homology, or the complementary sequence is there. So interestingly, only a part of the microRNA is complementary to its target. For example, here the lin4 microRNA has been shown, which is around 22 nucleotide is long, and this, it doesn't mean that the whole microRNA will go along with the MI risk complex and will go and bind to the target. Only a part of the some nucleotide remains, it goes and bind to them, rest will form this bulge. So that's the beauty, that a particular microRNA can silence more than one gene because they have a more than one target. Or in other word, I can say the 3' UTR region, they have a multiple binding site for the same microRNA, or they have multiple binding site for the multiple microRNA. That means a particular microRNA can silence more than one gene, or similarly, a particular transcript or gene can be silenced more than one microRNA. So these are some of the result of our study. These are the precursor structure of microRNA. This is basically novel microRNAs. So this is, since we are working on Zia maize, so that's why Z for Zia and MA for maize. So Zia maize is 39 and Zia maize 22. Here you can see this is the precursor microRNA, and this blue one is basically the microRNA star strand, and this is basically the passenger stand, and the red one, it would be the eventual mature microRNA that basically guide the silencing. So I mentioned that when the binding is imperfect, then it causes translation repression. So these are the 5 different mechanisms, either of which one can inhibit the translation. So the bound MI risk at the 3' UTR region, it basically compete for the cap binding site, or it may compete for the elongation factor, or it may compete for the 60S ribosome, or it may trigger deadylation, or it may induce the premature dropping of ribosome, or it may basically triggers both deadenylation and the decapping. So either of these mechanism, the imperfectly bound MI-RIS complex basically repress the translation of the target gene. So this is a single photograph showing the microRNA biogenesis, their formation, primary to precursor, then their movement, formation of MI risk, and their different mechanism of translation repression. So now there are certain differences between the animal microRNA biogenesis and the plant microRNA biogenesis. So as I mentioned, that in case of animal, primary microRNA is converted to the precursor microRNA, then it is transported from nucleus to the cytoplasm, where it is further processed by the dicer and microRNA duplex is formed. So in case of animal, the microRNA duplex is formed within the cytoplasm, but interestingly, up to this step, it happens within the nucleus only. So this is the microRNA biogenesis. And there is no DROSA-like protein. There are some dicer-like proteins which basically are involved in this processing. So there is one major difference, that miRNA duplex is formed in case of animal within the cytoplasm, but for plant, it is formed within the nucleus. There is no DROSA, there are dicer-like protein. And in case of animal, the binding of the MI risk to the 3' UTR region is basically the imperfect one, and it causes the translation repression, either of that 5 mentioned mechanism. But here, mostly the MI risk binds within the protein coding region of the target, and the binding is mostly perfect one, and that results in the cleavage of this target. So here the mode of inhibition is mRNA cleavage. Here it is mostly the translation inhibition. So in case of plant, mostly we found that microRNA cleavage, mRNA cleavage is there due to perfect pairing. But sometimes this plant MI-RIS complex, they can bind at the 3' UTR region and the 5' UTR region, and they can also induce the gene silencing through the translation repression. So this is the microRNA biogenesis in plant. I'm not going into details, but as I have mentioned, you see this is the microRNA duplex that is already formed within the nucleus. Then it is exported to the cytoplasm. Then agoven protein will bind, and that will form the MI-RIS complex. So until now, what we have understood, that there is a microRNA, and this microRNA has multiple targets. So it is obvious. So when because of this gene silencing, there is usually a negative correlation exists between a particular microRNA and their target. That means, for example, here, if under certain abiotic stress, if certain microRNAs, if a particular microRNA is upregulated under that particular stress, then it is expected that enhanced suppression of their target. It is obvious because there is a negative correlation. And this target, if it is a negative regulator of stress tolerance, that means under the stress, this upregulation of this particular microRNA, it basically induces that stress tolerance. Similarly, if certain microRNA under a stress is downregulated, so it is obvious there would be enhanced production of their target. And if that target is a positive regulator, then it will induce the stress tolerance. So what I'm trying to say is usually a negative correlation exists between a microRNA and its target. So this is MIR398, as Dr. Shati has already told. So MIR398, it has a very prominent role in abiotic stress, and this is basically a very important microRNA family. And this MIR398, it has a target of copper zinc superoxide dismutase, or CSD. So as I mentioned, this ascorbate glutathione cycle, the copper zinc SOD or the SOD, it basically acts as a first level of defense in plant because it scavenges the superoxide radical. So suppose under certain stress, this Mir398 is downregulated, which basically, what does it indicate? It basically indicates higher accumulation of their target, that is the CSD1 and CSD2, and subsequent protein would be formed. So since the copper zinc SOD or SOD is the scavenger of the ROS, that will basically give protection from the oxidative damages. That means a down regulation of Mir398 under a certain stress condition is basically give protection to the plant to overcome that particular stress. There is another example. Here you can see there are two very prominent microRNA, microRNA 160 and microRNA 167, and they have the target of these auxin response factor. This is basically a transcription factor. So suppose under certain stress, these microRNAs are upregulated. So it is obvious there would be a certain downregulation of the ARF. And these ARF transcription factor basically regulate the expression of auxin responsive gene, which are involved in the plant growth. So higher expression of Mir 160 and 167 under a certain stress would lead to the restriction in the plant growth. So what I want to say that whenever a plant is experiencing any kind of stress, one of the basic strategy that plant has adopted is restricted their plant growth. And this plant growth restriction is very much required to save the energy, because plants always try to save their energy, and that energy can be channelized to the defense purpose. And once the stress can be overcome or the stress period is over, the plant will regain its normal growth. So here the growth attenuation is basically a defense strategy of a plant to overcome the stress by upregulating the Mir 160 or Mir 167. So now I would like to share some of our results with you. So we did two different, today I will shed light on two different aspects. The first one is basically all about heavy metal stress. This is the identification and the characterization of the microRNAs in response to heavy metal stress, that is particularly the arsenic in maize. And another one is modulation of these microRNA profile under a submergent stress. So we all know this, the arsenic contamination of groundwater has become a serious threat to the sustainable agriculture in Southeast Asia, South and Southeast Asian countries, for example, India, our neighbor Bangladesh, and China. And there are different sources of this arsenic. So withering of rocks, minerals, and different anthropogenic activities, for example, indiscriminate use of these herbicide, pesticide, and chemical fertilizer, and also the soil irrigation with the contaminated groundwater. These are some of the prime source of arsenic in the soil, or you can say the groundwater. And in nature, arsenic basically prevails in two different forms. One is an inorganic form, another one is an organic form. Within the inorganic form, it may be an arsenate form or it may be an arsenite form. So this is a picture showing, you can see the worldwide situation. These are the red color, these are highly affected with the arsenic. These are the different subcontinents you can see. And here you can see our the map of India. So our state West Bengal, our neighboring state Bihar, Assam, Chhattisgarh, Jharkhand, Uttar Pradesh, even you see the Karnataka. These are arsenic affected areas, Indian states. And within West Bengal, the groundwater of so many blocks, it's around 80 blocks across the different districts, including our one, the Nodia, and Murshidabad, then Malda, then Bardhaman, and these North and South Chobbish Parganas, all are contaminated with the arsenic beyond the permission level. And the permission level is 0.01 to 0.05 ppm. So arsenate, that is the AS5, is basically enters the plant root system through the phosphate transporter, whereas the arsenite AS3, it enters the root system through silicon transporter or aquaporin. So once the arsenate it enters the plant root system, it readily reduced to the more toxic arsenite form, that is the AS3, with the help of arsenate reductase, you see, and in presence of the GSH. So once the arsenite is formed, it basically interacts with the sulfohydryl group of the enzyme and proteins, and which make them non-functional and thereby disturbing the normal cellular function. So we know that plant basically encounter these arsenic stress by mainly by two different mechanisms. One is basically a phytoceratin mediated detoxification pathway, and second one is by this modulating the antioxidant defense system to keep the ROS level within the limit. So phytoceratin, these are basically very low level, low molecular weight thiol peptide, and it is basically synthesized from the glutathione with the help of the PC synthase. And interestingly, the arsenate, that is the AS5, has no affinity for the sulfohydryl group present in the GSH or PC. So that's why only the arsenite form that forms complex with the PC and or the glutathione, whatever the form of chelation, then it get sequestered within the vacuole with the help of these ABC transporters. And these are the very two common plants of plant species that basically accumulate very high amount of arsenic from the nature. That's why these are known as arsenic hyperaccumulator. The first one is a very common Chinese break. It's Teres vittata, scientific name. You know, it's very common to us. And they basically accumulate very high amount of, and they can tolerate very high concentration of arsenic. And they have a very high root to shoot translocation of this arsenic, and it has been found that they sequester these arsenic phytoceratin complex or arsenic glutathione complex in the epidermal cell vacuoles of the frond. And studies have revealed that the young fronds with immature sori, they basically absorb more arsenic, around 3000 ppm, as compared to the fertile mature frond, which is around 1000 ppm. And apart from that, there is another very common plant, that is a Brassica Juncea, also known as Indian mustard or the brown mustard. They are also a good example of the arsenic hyperaccumulator. Since I have been working with maize, so maize basically, you know, this its scientific name is Zea maize, and it's belong to the family Poaceae, and is very important cereal crop. And it is basically the in India is basically the third most important cereal crop after rice and wheat, and it contributes nearly 9% in our national food basket. So these are table showing some of the metal responsive microRNAs. You see, these are the different microRNA families, MIR319, 171, 390, like these, and their involvement in these certain metal toxicity. So the first experiment that we did, it is all about the modulation of the microRNA expression profile in maize leaf under the arsenic stress. So we did this experiment with the following objective. The first one was doing the deep sequencing of small RNAs for identification and characterization of the arsenate responsive microRNA. The second one is the prediction of their potential targets. Third one was the validation of the microRNA as well as their target. And last one is to decipher the role of candidate microRNAs, which are involved in the gene regulatory networks associated with the arsenic stress. So this is basically the experimental flowchart. I'm not going into details, but I can tell you that what we did, we basically imposed that arsenate stress in the term in concentration of 50 micromolar arsenate to the 7 days old maize seedlings. And after 2 days of stress, we basically isolate the RNAs for downstream work, for example, for the smaller library preparation and the sequencing and all these things. So you may ask question why we have selected second day leaves, or why we selected or isolated RNA from the second day stress leaf. This is because during the pilot experiment, what we have seen that this second day onwards, there is a very sharp reduction in the shoot fresh wet. And also this histochemical staining with the NBT and Dept also revealed the oxidative burst, and that is very much prominent at the second day. That's the reason we isolated the RNA from the secondary leaf, from both control and the stress leaf. So these are the different steps of microRNA profiling, starting with the RNA isolation. Here you can see this is the control leaf, and from this is the stress leaf. You can clearly see the symptoms of arsenic toxicity. The yellowing and the drying has already started. So RNA isolation, then followed by the small RNA library preparation, followed by the deep sequencing using the Illumina platform, then bioinformatic analysis using a free software. This is basically a free software. This is UEA small RNA workbench, and we did the validation we validated the microRNAs, selected microRNAs with the stem loop QRT-PCR and their target with the normal QRT-PCR. So this is basically the overview of smaller library from plant samples. I'm not going into detail, starting from the RNA isolation through the quantification, then the check of redo, it should be around 7 to 10, and then smaller library preparation, then sequencing in a Illumina platform, or you can say you can use a HiSeq or MiSeq platform. So this is called the deep sequencing. Then this bioinformatic analysis, and finally we basically identify the known and novel microRNAs using the MeadProf and MeerKAT software that is already installed within this UAE software. And there is we did the prediction of plant microRNAs target, their potential target, using this PS RNA target analysis software. And BLAST2Go software was used to describe the biological process, molecular function, and the cellular components of the microRNA target, the targets of those microRNAs. So this is basically adapter removal tool. You can see these are some of the glimpses of that software that we used. So this adapter removal tool is basically installed within the UAE small workbench, and that has been used to print adapter. And there is a filter was used to discuss the low complexity reads. And this is a MeadProf, as I mentioned. It is used for the identification of known microRNAs. This is the Meer database that we use. This is the microRNA database, is Meer database. It's 22. Here you can see the version is 22. And these are some of the freely available major tools that can be used to study of plant microRNAs, their potential targets. So these are, you can see, these are Tapir is there, and Meer tool, Meer base, this Meer plant. These are the respective web links are there. And as I mentioned, MeerKAT was used for the novel microRNAs, for identification of novel microRNAs. These are PS RNA target that was used for the prediction of the microRNAs target in plants. The BLAST2Go software was used for the gene ontology study, description of the target this target mRNAs or target transcript. So this slide basically shows the size distribution of unique small RNA sequences in both the libraries. That is, the red one is the control one, and the green one is the arsenic stressed one. So in both the libraries, 24 nucleotide long units, these long unique small RNA sequences were found to be the most abundant, and this is followed by the 22 and the 21 nucleotide small RNAs. And this is basically the statistics of small RNA sequencing. I'm not going into details. So these are some of the figures showing the highest abundance of known microRNA families. Here you can see the Meer 166 has the highest number of members in both the libraries. Here the blue one is the control one, this is the arsenic stressed one. And you know that under when we impose the stress, arsenic stress, a lot of microRNAs were differentially modulated. What we found that in total 16 and 33 microRNAs, known microRNAs, or you can say concerned microRNAs, were found to be up and down regulated under the given stress condition. And you see, these are some of the examples of the up regulated microRNAs. This is Meer 8175 and their respective sequence, microRNA sequence is there. And this is the fold chain. That means with respect to the control, what is the fold chain, how much it increased, that has been shown here. So the expression of these particular microRNA, Meer 8175, which has a sequence of this, it basically upregulated as compared to the control. It is around 2.3-fold upregulation was there. And similarly, these are some of the examples of downregulated microRNA under our given stress condition. Here you can see Meer 166E3P. Basically, it shows the highest downregulation. That is, since it is minus, it means downregulation. So there is a 3.29-fold downregulation of that particular microRNA. We also identified some novel microRNAs, around 10 in number, and we categorize this as a ZMA, Z for the Zia and maize is a MA. So in total, 6 were upregulated, and 2 novel microRNAs were found to be downregulated, and 2 remain in a neutral state as compared to the control. So this table basically shows the predicted targets of conserved, or you can say the novel microRNAs. So here this is very important. This is particular microRNA. This is, you see, MIR156S, and it has the so many targets, and these are the putative function of those targets and the mode of cleavage. So after doing the bioinformatics, you can get all this information. And as I mentioned in the theoretical part, that a particular microRNA may bind or silence more than one gene. So you can see this MIR156S, it has so many targets. That means either of the target can be silenced with the particular microRNA, that is MIR156S. And these are the function of these targets. And these are the mode of inhibition. Here you can see this is cleavage. However, for this particular microRNA, we also got both type of cleavage and the translation repression mode. And most of the targets of these microRNAs are basically the transcription factor. You see GMYB is there, this is MYB is there, is Squamosha is there. So this is another this gene ontology analysis of these conserved microRNA target genes. You see this is MIR156A. There are so many targets, their description, and geodescription of cellular component. This is a nucleus based. Its location is nucleus, then molecular function is DNA binding proteins, and here there is no biological process, but for this particular microRNA you can see. So these are the different cellular components, molecular function, and biological process of the target genes or target mRNAs. So this is for the novel microRNA target genes. We did the similar gene ontology study. So what I want to say that under a given stress, a large number of microRNAs are basically differentially modulated. And what we found that these microRNAs, in our case, those are associated with diverse biological processes. For example, MIR156S, 166E3P, 166M, and 319B3P, all are associated with the plant growth and development. Similarly, these microRNAs are associated with the metabolic processes. Some, this is 167, it is basically involved in the ROS generation. These are some of the microRNAs which are involved in hormone signaling pathway, and these are the microRNAs which are involved in the microRNA biogenesis. So you can see that there are so many microRNAs which are up and down regulated under certain stress, and they are basically regulating diverse biological processes, which ultimately result in the stress tolerance, or you can say adaptation from that stress condition. So this is gene ontology categories. Upper panel is for the target gene for differentially expressed known microRNA. This is biological process, molecular function, and cellular component. This is for the novel microRNAs. Here what we did, we basically checked, we also validated basically the expression of selected microRNA. For example, after doing the bioinformatic analysis, you came to know that this particular microRNA MIR167D is basically down-regulated under your stress condition. That means this particular microRNA under stress condition is 2.4-fold down as compared to the control. But you have to validate this data. So what we did, we basically checked the expression of these particular microRNA using a stem loop QRT PCR. This is a special type of PCR. So using this stem loop QRT PCR, here you can find there also the MIR167 is down-regulated. That means here it is basically this QRT PCR or the stem loop QRT PCR data is basically validate our bioinformatics data or the deep sequencing data. Here also another example of MIR162B, which is down-regulated as per bioinformatics analysis, and it is also, in reality, it is also down-regulated. So these are all about the validation of the selected microRNAs. This is a similar picture where you can see the blue one is basically stem loop QRT-PCR study or the down regulation it shows, and these are the deep sequencing of that respective microRNA. So these are basically the validation of stem loop QRT-PCR along with the deep sequencing data. We also validated the expression of certain targets of those microRNA genes which are differentially modulated. For example, here you can see this is a GMYB, is a transcription factor. This is an auxin response factor I mentioned earlier. So you see these are down-regulated. Here you can see this is a significantly down-regulated under the stress condition. So we checked some selected microRNAs. We also checked the expression of their target just to validate the data. So we summarize our data in this form. This is basically a schematic illustration of summarizing all the microRNAs which are differentially modulated. Here you can see there are red arrow downward means down regulated and rest is up regulated. So here you can find that these are the differentially modulated microRNAs, and their these targets are there, respective targets are there, and these are their the different biological processes that they modulate. So finally, we proposed a hypothetical model. It is all about the role of two very important microRNA that we got interested. And this is MIR167D. It has this microRNA has the two target. One is the PLD alpha 1, that is phospholipase D alpha 1, and this is the auxin response factor. This is the ARF3. So you see, this is the down regulation of MIR167D. It basically indicates the higher accumulation of PLD alpha 1, and this PLD alpha 1 is basically responsible for the ROS generation. So this histochemical staining with the NBT and Dept, and also with the fluorescent dye, what we have seen, there is an oxidative burst. So basically, this oxidative burst is basically might be due to this down regulation of MIR167D, or you can see the upregulation of the PLD alpha 1. And auxin response factor, as I already mentioned, down regulation of AR3 is basically involved in the plant growth. So down regulation is basically it basically triggers the restricted plant growth under the stress condition. So similarly, there is another microRNA MIR319B 3P. It has two targets. Again, the GMYB is down regulated. I have shown in the last figure. You see here, you can see this GMYB is down regulated, and AR3 is also down regulated. So these are involved in the plant growth. So by upregulating these particular microRNAs, they basically the maize plant basically try to survive the stress by limiting their growth under the stress condition. So I'm not going details into the conclusion of our first experiment. I just want to share something. So that means we in total we isolated, you see, 22 upregulated and 35 downregulated microRNAs from the arsenic stress leaf. And you see this is quite a good number of microRNAs. That means you see that the 50 micromolar of arsenic exposure for the 2 day, it basically significantly affect the modulation or the expression of so many microRNAs. And apart from the known microRNAs, we also revealed, our study also revealed large number of novel microRNAs, which are exclusively expressed in the arsenic stress leaves, but whose putative targets are yet to be identified for better understanding their role in arsenic stress response. And recently in 2017, we have published this data, what I have shown in Environmental Experimental Botany. So if you are interested, you can go through this paper for more details. So now come to the second stress effect, that is again a very important stress, that is the submerged stress or the flooding stress. So flooding, it is mainly caused by the heavy metal or the heavy rainfall or the continuous rainfall in such areas where there is a poorly drained soil is present. So this kind of water stagnant condition, it basically affects the maize yield to some degree at almost all growth stages of the plant. However, the crop is most susceptible during the seedling stage. So whatever the condition, whether the plant is completely submerged or it is partially submerged, when the root is submerged under the water table, what it basically limits the water supply to the root. So this oxygen limitation under the submerged condition, it basically triggers a hypoxia stress or sometimes even anoxia stress, and this basically result in the metabolic shift from the aerobic respiration to the less efficient anaerobic fermentation. So this is our experiment. I would like to share this. Basically, you see, this is a control plant, is basically a 3-day-old maize seedling, and we imposed or we subjected these plants to complete submergence. And after the 4 days of submergence, you can find the growth is completely restricted, both the shoot growth and the root growth. So you see that the 4 days of stress, submergence stress, is so much deleterious for these young seedlings. So we also studied this conventional NBT dab staining with these fluorescent dye. So all these histochemical staining by NBT dab and the fluorescent staining with the DCFD and DHE all basically revealed that there is an oxidative burst under the flooding stress. So along with this, we also studied the expression level of the antioxidant enzyme. Here you can see this is the APX1. Under the submerged stress, there is a significant reduction in the APX1 transcript level, and similarly the copper zinc SOD, that is also significantly downregulated. And we also checked we also validate these APX1 transcript level with the what is called the Western blotting. Here you can see the very faint band as compared to the control. So this is all about the downregulation of these antioxidant genes, which might be responsible such oxidative burst that can be seen in terms of very deep brown color root tips, and this is deep blue color is NBT stain, and these DCFD after the staining of this DCFD or DHA. So we also studied the superoxide anion production within the tissue under the submerged condition as well as the control condition. Here you can see there is a very huge production, there is a very marked increase in the superoxide anion production under the stress condition. And we also studied the MDA level. Higher MDA means, as I mentioned, is basically indicates the higher membrane damage. And as I mentioned, when the root is under submerged condition, there is a limitation of the oxygen supply. So there is a metabolic shift from the aerobic respiration to the ethanolic fermentation. So you know that these are the three very basic enzymes which are responsible for the ethanol production or the ethanolic fermentation pathway. This is pyruvate decarboxylase, this is PDC, this is alcohol dehydrogenase ADH, and there is also the LDH, that is lactate dehydrogenase. So in our case, what we have found that we basically estimated the enzyme activities of these two enzymes, and in both the enzyme they exhibit a very high enzyme activity under the stress condition, which basically implies that yes, there is a metabolic shift from the aerobic to the anaerobic condition. So this is basically the graph showing the size distribution of unique small RNA sequences. Here also the 24 nucleotide long unique small RNA sequence was found to be highest, most abundant in both the control route and or you can say the submerged route, that is basically the this library, smaller libraries. And you see this is basically the Venn diagram showing some exclusively expressed and commonly expressed microRNA. Now you see this is this part is for the control route, this part is for the submerged route. Here you can see this common part, that means out of this common 188 expressed known microRNAs, 45 microRNAs were found to be differentially modulated. And most of the microRNA, very interestingly, out of these 45, 43 were found to be downregulated and 2 were found to be the upregulated in maize route upon submergence. So this is a statistics of small RNA sequences. I'm not going into details. This is a file, this photograph showing the highest abundance of known microRNAs. Here also you can find that MIR166 family has the highest number of members in both the families, both for control and the submerged route libraries. And here you can see these are the some predicted targets, predicted novel microRNA structures. As I mentioned, the blue one is a microRNA star and the red one is a mature microRNA. So here also what we have found that out of the 188 commonly expressed microRNAs, commonly means they are expressed in both control and submerged stress, only 45 are differentially modulated. And very interestingly, only these two microRNA, MIR169C3P and MIR393B3P, these two microRNAs were found to be upregulated under submerged stress, and all the rest 43 were downregulated. These are the predicted targets and gene ontology description, just I have mentioned earlier. So these are the gene ontology categories of biological process and molecular function. This is gene ontology categories for the novel microRNAs. This is for the upper one for biological process and molecular function. So summarizing our data, what we have revealed that the deep sequencing basically showed that the 5 to 4 known microRNAs and 48 novel microRNAs from the two libraries. There are huge number of microRNAs. are there, and there are very interestingly 48 were found to be novel. So, and most of these submargin responsive microRNAs, they were found to be associated with the diverse biological processes. Here again, associated with the plant growth and development, some hormone signaling, redox homeostasis, microRNA biogenesis, chromatin remodeling, and protein transport. So there are huge number of microRNAs which are differentially modulated, and they are basically involved in diverse biological process just to protect the plants, or just to, this is basically one kind of defense mechanism to survive under the stress condition. So here, as I mentioned in this theoretical part, that usually there is a negative correlation exists between a microRNA and its target. Here you can see we have done the stem loop QRT PCR for MIR160F5P. The expression is significantly downregulated, and its target was studied using the QRT PCR, that is the auxin response factor 8. Here you can find this is a significant increase. So downregulation of MIR160F5P with a concomitant increase in the ARF8, which is their target. So usually, as I mentioned, a negative correlation exists between the microRNA and its target. So similar negative correlation was found for the MIR172A and ERTF-RAP2-7 target. So we basically, this particular scheme showing the differentially modulated microRNAs, only the red two were found to be the upregulated, rest were downregulated. These are their respective microRNAs, and these are their respective the associated metabolic pathways that they are controlling. And finally, we proposed a hypothetical model. You see, this MIR172A, as I mentioned, it is downregulated, and we have shown, we have also validated this ERTF-RAP2.7 is upregulated. Now, this particular transcription factor basically triggers the expression of anaerobic genes. And we have checked that the level of the enzyme level activity of these ATH and PDC, which are basically the fermentation related enzyme, and those are upregulated. So that means here the upregulation of this particular transcript, or you can say the MIR172A mediated downregulation of this RAP2.7 is basically responsible for this conversion of the aerobic respiration to the ethanolic fermentation, and that leads to the restricted plant growth. Similarly, we have found there was oxidative burst under the submerged condition that was revealed after that histochemical staining and fluorescent dye, and the downregulation of these microRNA might be the consequence of that part. And so deregulation of the raw scavenging system. Here you can see that most of the antioxidants get downregulated. So the deregulation of raw scavenging system, that might be a determining factor for poor performance of those maize seedlings under the submerged condition. So there is another paper. If you are interested, you can go through that. It's very recently this year we published. This is all about the assessment of zinc oxide nanoparticle toxicity maize and integrative microaeronomic approach. If you are interested, you can go through that. Here you can find the modulation of microRNAs under a different group of stress. As I mentioned, it's a new class of pollutant, that is the nanoparticle stress. So this is my lab, plant stress and molecular biology labs. My students are there, the photographs of my students, and basically they are the pillars of my success. So if you are interested, you can join. So lastly, I would like to acknowledge all the funding agencies, both national and international, who basically supported my research. So, so with this, thank you. So thank you, everyone. So if you have any query, you can ask.
Thank you, Professor Hossein, for your excellent delivery. Really, it is superb. What to say more? It is superb. We are really impressed.
Basically, yeah.
Yes, you are right.
Basically, I have selected this topic. Yeah, basically, I have selected this topic because nowadays this particular microRNA chapter or the small RNA chapter, it is included in the MSc syllabus. So it's very new topic, and since I have been working in this field, so I think the MSc student, if they can understand better the mechanism what I have shown.
Definitely,
thank you.
the slides are much more informative, and so many MSc students are right now with us. So
Okay.
thank you once again. Now I am switching over to Dr. Subhadeep Chakraborty to conduct the interactive session. Dr. Chakraborty, please. Can you hear me, Dr. Chakraborty?
Yes. Am I audible?
Subhadeep,
Am I audible?
Dr. Chakraborty, can you hear me?
Am I audible? Am I
There is some
audible?
yes, there is some disturbance coming from. Please mute one set and go to only single set.
Okay.
Hello, Dr. Chakraborty.
Okay. So,
Yes, you are audible.
And thank you, Dr. Hossein, for your excellent delivery and very lucid presentation and very fun presentation. There is one question I found in the chat box by Dr. Tonda Bosha. That is, how can we address that the novel miRNA are associated with different metabolic process?
Sorry, I couldn't hear.
So
There is some problem
there
in.
was a question in the chat box by Dr. Tanmoy Bosha.
Okay.
That is, how can we address that the novel miRNA are associated with different metabolic process?
Yeah, you see, I have mentioned it is all about this. After doing the bioinformatic analysis, we have seen that these microRNA, these different biological processes are associated. But I have mentioned in my conclusion part that their target should be validated, and then we can come to know that whether these novel microRNAs are actually the target are actually those microRNAs, and those biological processes are involved. So whatever I have shown, it's basically the prediction. So for that, one has to do a race, that is 5' race, to exactly locate the target site. Or you can say that after doing this part, one can validate, and they can tell that yes, these novel microRNAs are associated with the certain targets, and they are associated with the different metabolic pathways.
I have one question, that is there any component of microRNA for different factors of stress maker? Is there any
Sorry.
commonality?
Sorry, Dr. Chakraborty, you are not
I,
audible clearly.
yeah, I could not
We can't
hear.
hear you clearly.
1 minute, please. 1 minute. I think there is some problem. So can you hear me?
Dr. Chakraborty, you are not audible clearly.
Not audible.
I think for technical reason you have to use two sets. It is better if you mute one set.
Now am I audible?
Yes,
Yeah.
yes, now you
Yeah.
are clearly audible. Okay.
Yeah.
Yes. So my question is that is there any kind of commonality between the stress factor, suppose cadmium toxicity or arsenic toxicity or nanoparticle toxicity of the miRNA?
Yeah. There are.
So that miRNA can play a role as a biomarker.
I'm trying to give your answer the first part. We used to find that there are several known microRNAs which are commonly expressed. If suppose, for example, MIR398, this is a very its target is copper zinc SOD. So in most of the cases, what we have found, whether the stress is a drought or it may be a flooding stress or it may be a nanoparticle stress, so you can get a 398 whether it is upregulated or downregulated, because these are conserved microRNA or the known microRNA, because these are evolutionarily conserved throughout the plant species. And regarding the 398 or a particular microRNA, you cannot use as a marker that expression of this particular 398 means that it's salt stress or like that. These are common. These are known microRNAs. So these you can find it in different plant species under different stress conditions.
Okay. And one of your slide, one of your slides showing that the microRNA related to ROS generation
Yeah.
or metabolic processes and hormone
Yeah.
regulation involved.
Yeah.
So,
Yeah.
which hormones mainly are which hormone
Basically,
microRNA?
yeah,
Which
yeah,
particular hormone?
Basically, what we have found, I didn't include those slides because of the time constraint.
Yeah.
What we have found that there are a lot of auxin hormones are basically regulated, and
Okay.
this is 2,4-D was there, then IA was there. So these
Okay.
hormones were basically what we found is basically downregulated. So these auxin responsive factor ARF, these are basically the transcription factor, and they are involved in
Okay.
the regulation of these auxin responsive genes. So ARF, once the ARF is down regulated,
Okay.
it basically indicates there would be a
Yeah.
less production, and that basically you can say suppress the growth. So as I mentioned that
Okay.
growth suppression is basically you can say this is a adaptive feature of the plant to withstand the stress period. So once the stress period is over, the plant will try to regain its normal growth. So
Okay.
mostly auxins,
And
mostly
I have
auxins.
another. Mostly okay, thank you. And I have another more question, please. That is, you have also shown that the miRNA is related to ROS generation. So ROS
Yeah.
generation ultimately leads to apoptosis. So have you found any kind of relation with apoptosis also?
No, we didn't study the apoptic pathway. It is more common in animal system, but however in plant system also apoptic pathway is there. But we didn't study that one. But what we have studied that the effect of the toxic effect of ROS on in terms of a membrane integrity, or you can say the cell viability, all these parameters we have checked. Okay,
Okay.
we didn't
And
check that.
the last one. Okay. And the last one is what should I say? Farmers are mainly affected. How these results can help the farmers?
Basically, this is a very pertinent question. Basically, this is what our result is. Basically, you can say it's a basic information that we are providing to the scientists who are working with the who are basically the genetic engineer. So, what we have published the data is the up and down regulation of the particular target microRNAs, or you can say the candid microRNAs. So if a genetic engineer, they can design and crop by overexpressing that particular candid microRNA or downregulation of that particular microRNA, it would be beneficial for them. So our result basically provides a basic information to the scientists who are working with the GM crop or the transgenic.
Okay, so thank you, Dr. Hussain, for your nice delivery and lecture. So I am now switching over the microphone to the Kaushik Sengupta for the valedictory session. Kaushik, please.
Thank you, Dr. Chakraborty. Okay, I'm continuing. Before entering into the valedictory session, Professor Hoshin, one information is required from my end. May I ask you?
Yeah, please.
Actually, in your slide, you mentioned the broad spectrum target of 156 miRNA BS, 156 BS. Can you hear me?
156, okay. Yeah.
My question is very basic question. One, is it act similarly in case of both monocot and dicot?
Since I have been working with maize, particularly maize.
Oh, I see.
Yeah, but since these are known microRNAs or the conserved microRNAs, I hope that will have the same target. Yeah, but the modulation, whether it would be down or up regulated, it will vary depending on the stress. Okay, whether also it varies from species to species as well as from monocot to dicot. But what I can assure that mostly the target would be same because it's a conserved microRNA. This is evolutionary conserved throughout the plant species. So a MER 156, the sequence of MER 156 For example, in Arabidopsis,
Yes.
also in maize and also in rice, the sequence remain almost same, and these are called orthologous sequence or ortholog. So what I can assure, the target remains same. However, the modulation, it depends on the plant species, the condition, the stress treatment, and all these things.
Thank you, Professor Hussain.
Thank you.
You have nicely answered.
Thank you.
One thing, it is coming in my notice that our respected professor is continuously with us, though
Yeah,
he is
thank you.
from different discipline, thus with continuous effort and concentration he is with us. So thank you, sir, once again for being with us continuously. And now this is a high time that I am requesting Dr. Bhaskar Chatterjee, our enthusiastic and dynamic coordinator of IQAC, to place his vote of thanks on behalf of Navadip Vidyasagar College. Dr. Chatterjee, please proceed.
Hello, am I audible?
Yes, you are clearly
Hello.
audible.
Yeah, okay. Thank you, Kaushikda, and good afternoon, everybody. First of all, I would like to congratulate the Department of Botany for organizing such a nice webinar. And at this point, I took the opportunity to offer my sincere thanks to our distinguished speaker for the day, Dr. Zahid Hussain, for such an excellent and lucid presentation on today's topic, that is microRNAs, key players of plant responses to abiotic stress. I thank him on behalf of IQAC and also from Navadip Vidyasagar College for accepting our invitation and being with us today. And I would also like to thank all of our participants who have come forward and participated in this webinar to make it successful. I would like to thank our principal, sir, Dr. Shrapan Kumar Roy, and our president of the governing body, Mr. Biman Krishna Shah, for their all-time support and encouragement. Finally, I thank our organizers of this today's webinar, all the faculty members of the Department of Botany, convener Dr. Shatida Shur, and all others who have supported us with the technical issues, Professor Dipanjan Ghosh, Sri Amalendu Das, and Professor Rajkumar Mondol, and whose combined efforts today have made this webinar successful. So I finally thank all of you and thank you for your patience. Thank you.
Thank you. Thank you.
Thank you, Dr. Chatterjee. We are really at the final end. But all participants, may I draw your attention, please? You will get the feedback form link in your message box. Kindly, carefully fill it up and then submit to get the e-certificates. So this is for the day. Please stay at home. Be safe. Thank you. Good. Have a nice day.
Thank you.
Thank you.
Thank you.